Okay, so here we're going to start out with the different experiments that led to our understanding that DNA is the model of inheritance. So here with, if we look back at the 1908, there was a guy named T.H. Morgan who was studying, I know it looks like a butterfly, but he was studying the fruit flies, Drosophila, and he figured out, oh my gosh, traits and like our phenotypes, like who we are, is inherited on chromosomes. Now, chromosomes are made out of DNA and protein. If you recall, we have these things called histone proteins. So here we have histone proteins, and then our DNA is like wrapped around histone proteins. Anyway, so when we look at the quest to understand which molecule is the molecule of inheritance, was it DNA or was it protein, there's a series of experiments. And in 1928, there was a guy named Frederick Griffith who was working on the transformation. He was working on figuring out a cure for pneumonia. So he worked with living non-pathogenic bacteria. So this NP stands for non-pathogenic. And those ones had a rough appearance. And then there was pathogenic bacteria who had a capsule around their outside. And that capsule is what helped them kind of evade the host's immune system. And that allowed them to live long enough to infect. So what he did with his experiment is he infected mice with non-pathogenic bacteria, and the mice lived. He injected the mice with pathogenic bacteria, and they died. Then he took some of this pathogenic bacteria that he killed with heat. So these are dead, formerly pathogenic bacteria. When he infected them into mice, the mice survived. Now here was the trippy part, is he took these dead pathogenic bacteria and these living bacteria that were non-pathogenic, and he added them together. And it turns out that the mice died. And so here Griffith was like, oh my gosh, something was being passed from this dead pathogenic bacteria that on its own does not cause disease. It was being passed to the non-pathogenic, and then they became pathogenic. They became disease-causing. So somehow these non-pathogenic bacteria were transformed to become pathogenic. So this was Griffith's transformation experiment. Now back in the day, 1928, he didn't know what was the transforming factor. He didn't know what was doing the transformation, but something changed its phenotype. And that brings us to McCarty and MacLeod, who 16 years later were trying to figure out, well, what was the transforming factor? Was it DNA, RNA, or a protein? So McCarty MacLeod used enzymes to isolate the DNA, RNA, and proteins that you would find. So they were working with this dead pathogenic bacteria. They were treating this bacterium with enzymes like DNase. DNase breaks down DNA, protease breaks down proteins, and RNase breaks down RNA. So when you treat these dead pathogenic bacteria with DNase, you would leave them with working protein or working RNA. If you treat them with protease, you'd leave them with functioning DNA and functioning RNA. If you treat them with RNase, you destroy the RNA, leaving them with DNA and protein. So they treated the dead pathogenic bacteria with these enzymes to see which ones still caused transformation. So they wanted to see if you had this, what is going to cause the non-pathogenic bacterium to be transformed to become pathogenic, right? Like which molecule? Is it the DNA that was being passed to cause the non-pathogenic to become pathogenic? Was it protein? Was it RNA? And basically, by isolating each of these molecules, they were able to confirm that DNA is what's is the transforming factor, causing a change in genes and therefore the phenotype. So when the dead pathogenic bacterium, it basically passed its DNA. It didn't pass, it was dead. But this bacterium here, the non-pathogenic one, was able to take up the DNA from the environment and incorporate those new genes into its chromosome. And then those new genes cause transformation and a new phenotype. Okay, then we have Chargaff. So now that we know the DNA is a molecule of inheritance, that leads us to Chargaff's rules, where you have, he figured out that, because at this point we know DNA is a molecule, but we don't know if it's like what it looks like. So Chargaff figured out that the amount of A in a DNA strand equals the amount of T, the amount of C equals the amount of G, and that's what we call base pairing. So that was 1947. And then we have 1952, 51, Hershey and Chase, as happening around the same time as like Watson and Crick and Rosalind Franklin. So Hershey and Chase, though, they wanted to kind of confirm DNA as a molecule of inheritance. They were curious if viruses also use DNA as their main molecule of inheritance, because all viruses are made out of a protein coat and DNA or RNA. And so they used these bacteriophage viruses where they made radioactive phosphorus on the DNA with radioactive phosphorus, because when we look at a nucleotide of DNA, it's this phosphorus here that they made radioactive. And then the protein coat has sulfur in some of the amino acids. So they grew phages that were radioactive for phosphorus and sulfur, and they allowed the phages to infect the bacterium. Now, they didn't know which part of the bacteriophage actually entered into the bacteria in order to give the directions for virus replication. And so what they did is they allowed the phages to infect, and then they spun the infected bacteria and the phages in a blender. And the reason for this is because the heavier bacteria will sink towards the bottom. So if, like, here's the liquid, well, then the heavier stuff will sink. And now this kind of separates anything that did not enter the bacterium from what did. So here you would have, or what they found was those protein coats with the radioactive sulfur were floating in the, those are S's, sorry. Radioactive sulfur were floating in the supernatant. Where down, they actually found radioactive phosphorus within the bacteria down at the bottom of the blender. So here, the fact that they found radioactive phosphorus within the E. coli cells basically confirms, wow, DNA is the molecule of inheritance for viruses as well. That's what they use as their genetic material. And then we have Hershey and Chase, not Hershey and Chase, we have the famous photo 51. Rosalind Franklin was working on X-rays, X-ray crystallography on the molecule of DNA. And basically, with this, any person at the time who knew a lot about how to read X-rays of molecules could look at this X here and know that it was basically a double helix, but like looking down at a double helix. So Watson and Crick got a hold of this photo 51, and one of them knew, oh my gosh, it's a double helix. And using Chargaff's rules that A equals T and C equals G, and the idea that it's a double helix, they were able to build the model, a three-dimensional model of DNA, and basically get the credit for discovering the structure. Now there's actually one more experiment that I'm not mentioning in this video, and that's Messelson and Stahl, and they're the ones who figured out how this double helix replicates. They figured out semi-conservative replication of DNA, but we'll cover that at a later time.